What's up, baby penguins? So I am live in my closet so that I can do this Unit 5 or CED Unit 7 AP review. As every single time, every month, well, actually, before each of my students have their unit exams, I come on live just to do a quick session for my students. I save it on this account just so that if, whenever you're taking this unit, logically, you'd be able to, watch this live stream if you're needing to. Wow, there's a lot of people that just logged on. Good evening, everyone. Unit 5 is all about evolution. And so I have a quick little PowerPoint like I normally do. And then I have 2 FRQs. One of them is a short FRQ that we're going to go over, and then one of them is a long FRQ that we're going to go over. And if you have questions about Unit 5, which is evolution, which, again, I'm doing my students Unit 5, which for everyone else, that's CED Unit 7. So if you have questions, pop them down in the chat. I'll answer them as we go. But again, this is solely and completely on evolution. And yes, I'm in my closet because my child is sleeping. The things that we're going to talk about, we'll talk about Hardy-Weinberg. We'll talk about the biological species concept. We'll talk about reproductive barriers, and we'll talk about selection. I noticed as I was getting ready for the session that I left out the evidence of evolution, but there's, the evidence of evolution is very obvious and mostly just looks at the homologous structures and looks at, the convergent evolution versus, of course, evolution. Convergent evolution deals with that they have the same structure due to they were in the same environment. So they're not actually related to each other versus when we look at just our natural selection, our modification, descendant with modification, we're looking at that they had a common ancestor. Okay. And so all the evidence of evolution is just looking at looking at the fossil evidence, looking at molecular evidence. Of course, we know that DNA is better than proteins when we're looking at that molecular evidence. And it has to do with the fact that DNA is going to show a lot more of this variability in it, right? So if you can have a change in your nucleotide structure, you might not see it in the proteins. Why? It could be in your intron. And we all know that the introns are cut out during post-transcriptional modification. Right? It could be a silent mutation. And we remember that silent mutation means that it's still going to code for the same amino acid. So when we're looking at what's better between DNA and protein, DNA is going to be much more accurate to show the amount of differences that have occurred in organisms. Okay. Wow. I can't believe I have this many people in the live. Makes me so exciting. All right. So let's get started. So real quick, Hardy-Weinberg. Okay, Hardy-Weinberg's big thing is its equilibrium, and it's to say whether evolution has occurred or not. Okay, so there's 5 fingers of evolution, and this did not come about me. So Paul Anderson from Boson Biology, he did a TED Ed video, and he goes over this really easy way to remember the 5 conditions of Hardy-Weinberg. Okay, so your pinky finger is going to represent that we need an extremely large population size. Extremely large population size because if a random thing happens, genetic drift, a random thing happens, it could cause certain alleles to become fixed. It could significantly reduce your population. And so the new population may not be representative of the initial population. And so genetic drift affects small populations more than it affects large populations. So we need there to be an extremely large population size. And hey, look. So the ring finger represents that we need to have random mating. And sometimes I'm not wearing my wedding band when I do this, and it always just makes me laugh when I'm actually wearing it because I'm bad at my ring. Anyways, so random mating. We can't have any type of sexual selection. So we all know the blue-footed booby and how the blue-footed booby shows off its feet, to show, okay, I've got the amazing blue feet. You want to mate with me because my feet are the most bluest feet you've ever seen. And I've got the good genes, right? So we can't have any of that taking place. We can't have any of these mating rituals that demonstrate that one is a more favorable trait than another. I've got the most spots on my guppy fish. No, we can't have any of that. Okay. The middle finger represents mutations. We can have no mutations. We want there to be any mutations because the mutation is going to add a new allele, which is of course automatically going to change my allele frequency. And then of course we can't have any gene flow. So we can't have any immigration. We can't have any immigration. Nothing can come in. Nothing can go out. And so again, we're looking at ensuring that our allele frequencies stay the same. Okay, because that's all Hardy-Weinberg's looking at is, do the alleles change? And when I'm looking at these alleles, I'm talking about, do the genotype frequency change and do the phenotype, the genotype. So we were looking at the alleles, we're looking at the genes. We're determining that they basically are going to stay the same from generation to generation to generation to generation. For hundreds and thousands of years, these alleles stay the same. And we all know that's not happening, but these are the conditions that we need to have in order to make that. And the last thing is your thumb. No natural selection. No trait can be favorable or unfavorable for an organism, okay? So we need there to be no natural selection because natural selection is, of course, going to select for those favorable traits, which will increase that allele frequency in the population. So I've got a little picture to help you out. So we got that. Okay, so again, the 5 fingers of evolution. If you have not watched this video, it's literally like a 3-minute video. Just go to Google, type in the 5 fingers of evolution. It is a TED Ed video. Paul Anderson, you'll recognize his voice. And it's honestly has great graphics. It's really a good video. So as I said, extremely large population size. We need there to be random mating, no mutations, no gene flow, and no natural selection. And if all these conditions are met, then that means our allele frequency is not going to change and our population is not evolving. Okay. So there's 2 different equations that we look at when we talk about Hardy-Weinberg equilibrium. Okay, so on my screen, I've got the main one. Okay, so P squared plus 2PQ plus Q squared equals 1. Okay, in which the P is going to look at your dominant allele. So if I have P squared, that means that I have 2 dominant alleles, and we all know that 2 dominant alleles means it's homozygous dominant, right? So P squared is your frequency of your homozygous dominant. Okay. Q squared, so we said that Q is your recessive allele. And so if you have 2 recessive alleles, we know that is homozygous recessive. So Q squared, Q squared is our homozygous recessive, okay? And then if you have 1 P and 1 Q, you have 1 dominant allele and 1 recessive allele. So of course, 2PQ represents my homozygous genotype, okay? And so this equation is just looking at there being 2 total alleles in population. Now, this formula gets completely more complicated when you up a notch, right? So, for example, I can't remember what exam it was, that I think that they may have given you the formula for one where it's 3 alleles in the population, okay? And they're asking you to solve for one of them, okay? Now, on that question, it was definitely way more complicated than it should have been. Okay. So they're asking, I believe, if I remember correctly, it's like a blood typing problem. And so what you have to do is you have to say, what is the frequency of the recessive allele, of the little I, right? So all you're going to have to do on this is take the individuals that have O-type blood and just do that square root and you'll have your answer. Hint, hint, nudge, nudge to my penguins that are watching right now that have their test tomorrow. Just take the square root of your O and you'll be perfectly fine. Okay, so there's 2 main ways that we calculate this. Number 1, we look at that formula that I just mentioned, the P squared plus 2PQ plus Q squared equals 1. We start out with our Q squared and we solve using Q squared, come into Q. Then we find our P, then we find our P squared, then we find our 2PQ. It's just literally doing square roots and squaring them and you can solve all the problems, okay? So now you can only do this, okay, if you're only given some of the phenotypes, if you're only giving some of the allele information. If you're not given all of the information, you don't solve it that way, okay? So if you are given, you have this many that are homozygous dominant, you're given this many that are heterozygous, and you're given this many that are homozygous recessive, you need to solve it with the allele method. Okay, so if you've done the HHMI malaria problem, which my students had to do as their big lab that they had to do 2 Fridays ago, I'm still grading, in which you calculate it by taking the number of homozygous dominant and you multiply by 2. Why? Because homozygous dominants have 2 dominant alleles. Okay, so homozygous dominant times 2. And then you're going to add in the number that are heterozygous. Okay, because there's only 1 dominant allele there. And then you divide by the total number of alleles of the population, and that will give you your frequency of your dominant allele. You do the exact same thing in terms of your recessive allele. So we take the number that are homozygous recessive and we multiply by 2 because they have 2 recessive alleles. And then you're going to add the number that are going to be heterozygous. Okay, and that will give you the number of recessive alleles divided by your total alleles, which gives you your frequency of alleles. Okay, so this is kind of how we have to solve those problems because we've been given the number of alleles. So we're not doing pedigrees right now because this is only looking at evolution. So you can ask that question, not on my live. Okay, so here is what I just mentioned. Okay. So we're given the number of all the individuals of each genotype calculated by counting the alleles. Okay. So what I just mentioned, if you're given those different individuals, you have to count your alleles. Okay. If you're not given the number of individuals in each genotype, then you'll calculate using Hardy-Weinberg and you'll start out using Q squared. Okay. That's really comes down to is using that Q squared. And as I said, once you find Q squared, you do the square root of that, which gives you Q. If you have Q, you then find P, and then you can do P squared, and then do 2PQ. Okay, easy peasy. Okay, so biological species concept is the main concept that we're going to use when we determine if 2 individuals are in the same species. Okay, so the big thing is, are they able to interbreed? So can they mate together? Can they come in contact? Will they attempt to mate? And if they do mate, if they're able to interbreed, do they produce fertile and viable offspring? Okay, so that is the big thing, is that biological species concept states that they can interbreed and produce fertile, viable offspring. So we have all these reproductive barriers that are going to have the goal of inhibiting them from trying to interbreed and inhibiting them from having fertile, viable offspring. If there are 2 different species, they should not be mating and they should not be having new, hybrids, right? So we have a couple different barriers. Okay, so they're called pre-zygotic barriers and post-zygotic barriers. A pre-zygotic barrier means that it's taking place before the zygote is formed. So these are things that are going to inhibit them from interbreeding. Post-zygotic barriers mean that they're taking place after the zygote is formed. Okay, so these are going to inhibit them from being viable and inhibit them from being fertile. Okay, so this is when we look at these different things. So let's look here. You see, we have our pre-zygotic barriers and we have our post-zygotic barriers. I have a picture of them. Wait, I lied. Sorry. So these are all our pre-zygotic barriers. Okay. So the first one you have is habitat isolation. Basically, they live in 2 different habitats. So since they live in different habitats, they're not going to mate with each other because they live in 2 different habitats. A big example that I've seen for a couple of years is there is a hawthorn fly and there's an apple fly. And so since one lives in the hawthorn tree, one of them lives in the apple tree, they don't ever come in contact with each other. So they don't mate. Okay, so again, we're inhibiting from interbreeding. Okay, another one you could have is temporal isolation. This has to do with that they mate at different times. Okay, so we have the eastern skunk and the western skunk. One of them mates during the summer and one of them mates during the winter. So since they mate at different times, they don't mate with each other. Okay, so again, we're inhibiting interbreeding. Another one you can have is our behavioral isolation. So there's our beautiful blue-footed boobies. My students know how much I love the blue-footed boobies. And so what they do is they do this dance and they show off their feet, their beautiful blue feet. And when they show off these beautiful blue feet, it then attracts a mate. So if you have a different dance, then you're not going to be able to attract that mate. Okay, so behavioral isolation has to do with that they have a different mating ritual. So it could be a song, it could be a dance, it could be some type of ritualistic activity that these animals do to attract their mate. And if they have a different one, then they're not going to mate with each other. Okay. So we also have that they've now attempted to mate, right? They're going to try to mate. And this gives us our mechanical isolation. So the best way to state this is talking about that we have some type of anatomical difference. And so because of the anatomical difference, the 2 are unable to actually mate with each other. Okay. Example I saw the other day was about plants, right? And so the sperm, the pollen, doesn't cause the pollen tube to germinate to allow for the egg to be fertilized, okay? And so that right there is mechanical isolation. The example we saw on the slide a second ago was the snails. One of them has a clockwise shell, one of them has a counterclockwise shell, which caused them not to be able to orient with each other to be able to mate, okay? So mechanical isolation is anatomical differences, okay? And the last one we have for pre-zygotic is our gametic isolation. Gametic isolation has to do with that the gametes don't fuse, okay? So we have these glycoproteins that are on the membrane, and so that glycoprotein we know is part of signal transduction, and so maybe they just don't have the appropriate receptor, and thus the sperm and egg don't fuse, and so if they can't fuse together, then their nuclei are unable to fuse together. Thus, we don't make the zygote. So all of these we're looking at are pre-zygotic barriers. So, or is habitat isolation, temporal isolation, behavioral isolation, mechanical isolation, and gametic isolation. You can state geographic isolation that kind of falls within habitat isolation. Those are kind of more or less one of the same, where geographic isolation just looks at that there's some type of geographical barrier, whether it's a mountain range, whether it's a lake, an ocean, or whatever that's separating these two organisms from mating. Okay. So post-zygotic barriers we can have, there's 3 of them. Okay. We can have reduced hybrid viability. Basically, the organism is not viable. It's not healthy. It's got a lot of medical problems. It's just not doing so good. It's not viable. It's not, it's very feeble. It's weak. It's got problems. Okay. You can have reduced hybrid fertility, which has to do with that the hybrid is now sterile. Okay, so we said before, they're the same species. They produce viable, fertile offspring. So with the horse and the donkey, they have an uneven number of chromosomes. So I cannot remember the number. It's really not important. But because of the fact that the horse and the donkey don't have the same number of chromosomes, when they fuse together, the mule has an odd number of chromosomes. And we all know during meiosis, you need to have the homologous chromosomes pairing up in order to produce your sperm and your egg. Well, they have an odd number of chromosomes, so they can't pair up. Thus, they are unable to make the sperm and the egg, and they are sterile. And the last one we have is reduced hybrid breakdown. So our first generation, they're strong, they're awesome, they're fit, there's no issues, they're great. But the more that time progresses, then we're going to start to see them break down. We're going to start to see them become weeble. We'll start to see them become weak and feeble. They're going to be not fit. They may become sterile. And so that's as we go through multiple generations of these hybrids, they're just going to start breaking down. And again, the goal of the post-zygotic barriers are to inhibit further hybrids from forming, right? Because in order to be the same species, they can interbreed and produce fertile, viable offspring. So all these barriers are inhibiting us from making those hybrids, okay? So then how do new species come about? So that's something called speciation, okay? So there's 2 different types of speciation. You have allopatric speciation and you have sympatric speciation. Allopatric has to do with that they're apart. There's some type of geographical barrier. There's something that's separating these organisms from each other. And because they're separate, they're going to diverge. There's a mutation that will occur in one population that doesn't occur in the other. There's natural selection taking place. There's something that's happening to these populations while they're separated, that when they come back together, when that geographical barrier is no longer there, when they're able to come back together, then they can't mate together because there's been so many differences that now they're 2 different species. Okay. They may now have different, they may now have mechanical isolation. They may have, biological isolation. They now have any of those isolations we just talked about can be the reasons why they don't mate anymore. Okay. Sympatric has to do with that they're in the same area. Okay. So there's something that inhibits them from mating. Okay. Whether the water got cloudy in one area and they just couldn't see their mate, and so they stopped mating and then they diverge. These are just basically something that's happening within the same area. Habitat isolation is a great example of this. So although the hawthorn flies and the apple flies were able to kind of intermix, the hawthorn flies chose the hawthorn tree. The apple flies chose the apple tree. So although they were originally the same population, they've now diverged from each other and become 2 different species because now they have habitat isolation. They're in two different habitats and they're not mating with each other. So now that they do come together, they're not going to mate with each other because of all the other barriers we talked about. Okay. So with natural selection, we're going to see that there's kind of three things that will happen. Okay. So let's look at the different selections. You can have directional selection, which has to do with that over time, they're going to move in a direction. Okay. You could have disruptive selection where we're going to disturb this intermediate and we're going to kind of just see the two extremes. And then you have stabilizing selection, which is where we're going to stabilize in that middle. Okay. So with directional selection, we said that it's going to move in a direction. The peppered moth experiment, or actually one experiment, but the peppered moths are a great example of this, right? So originally the moths were very light. Okay. They blended in with the trees. They were spotted, all that fun stuff. And then of course we started getting all of this industry that started putting gunk in the air, right? So the gunk built up on the bark of these different trees. And so the lighter shaded pepper moths were able to be seen. And so they were, preyed upon. And so there was a dark allele within the population. And the individuals that were darker were more likely to blend in and camouflage. So they were less likely to be eaten. And so we saw an increase in the dark allele in the population. So we saw a directional selection. They moved from being light to being dark. And now, of course, we've got the clean air, all these acts are going in. So, of course, it has swifted back. But again, directional selection is just looking at that it's moving in a direction. Okay. We said that we also have disruptive selection. This is going to be picking up the two extremes and dropping the middle. Okay. So there's something that's selecting for those two extremes. The best example I can come up with is the finches. So Darwin's finches, there was a drought. And because of the drought, all that were available were these really hard seeds and then these little insects, right? So the finches that had the little itty bitty beak could get to those little bugs, right? And the finches that had the big beaks could get to those hard shells and crack them open and they could get food. But those birds that had the medium beak, they could now compete with the small beak to get the insects, and they couldn't crack the shells open to get the seeds. So they died. So we saw a decrease in the medium-beaked bird, and an increase in the small, and an increase in the large-beaked bird. And so that would be disruptive selection. So we see the increase of the two extremes and a decrease of our intermediate. Okay. And last but not least, we could have stabilizing selection. Stabilizing selection is going to, of course, stabilize that middle. Okay. So there must be something that happens at the low end and something that happens at the high end that is selected against. Okay. Good example for this is birth weights. Most babies are kind of in that 5 to 7 ballpark. If the baby's too small, and we're thinking evolution as in long time, before modern medicine, the small babies probably didn't survive because they were so little, right? And the large babies, kind of had trouble. So they also didn't survive. And so we have that 5 to 7 ballpark is where most babies, could survive. They were viable. They were able to get out with no trouble. And so we started to see that that's kind of where we've stabilized. I saw another example looking at the number of, eggs in a nest, that usually they're in a 4 to 6 ballpark because of that's just natural selection is selected for having 4 to 6. I guess they can take best care of them and then it's just not too many, all that good stuff. So what is natural selection? So natural selection has to do with that there is a trait and this trait exists in the population. That's a big problem that students have is that they sit there and think, oh, well, the antibiotic caused the resistance in the bacteria. Well, no, it didn't. There's a trait already in the population. And then because of something, we see that there is a selection for that trait. And so that trait allows this individual to be more likely to survive and more likely to leave more offspring. Okay, and those offspring, of course, can survive. Fitness has to do with your contribution to the gene pool. So the more offspring that you have, and the more that those offspring are able to continue to have offspring, the more fit you are. And that's what we're looking at with natural selection, is which individual is most fit. So be very careful about stating about fitness and survival of the fittest, because that's a really bad word. Like, it's just like, mitochondria is the powerhouse of the cell. Nails on a chalkboard. Bad. Okay. So never say that, survival of the fittest. We're always talking about that the individual is more likely to survive and that they're able to leave more offspring. Okay, and we're talking about the individual with the trait, but we see that overall that the population is changing. Okay, so let's look at our little example. So here we see that I have these, I don't know what they are. Some type of critter. They're beetles. Okay, so these beetles, there's black beetles and there's white beetles. Of course, the white beetles can be seen on this dark rock, and so they're traditionally eaten by the bird. Okay, and so the ones that survived are able to pass on their traits, okay? I mean, if you notice, these are all heterozygous. We have a homozygous dominant there, but most of these are heterozygous, okay? And so in our next generation, we see that there are still some white of these beetles, but most of the population is becoming black beetles. And you can see that the allele frequency has changed. So we went from having a dominant allele frequency of 0.3 to having a dominant allele frequency of 0.6. So again, we're seeing the allele frequency changing, which is showing us that evolution is taking place, okay? And if we continue seeing this taking place in the population, we're going to see more and more of those dark beetles in the population. So looking at the natural selection part of it, right? So these beetles were more likely to survive, and they had more offspring. So we're seeing an increase in the dark allele in the population, okay? So that is our quick review of the concepts. And there's something, nope, that's a question from my story. There's no actual question in the box. Okay. So I've got 2 FRQs to go over with y'all. I've got the short FRQ and then, of course, the long FRQ that we did in class today. That way we have that moment to kind of go over it. Okay. So the short FRQ that we have is 2019 and it's number 5. Okay. So a researcher studying the evolutionary relationship among 5 primate species obtained data from a sequence of mitochondrial DNA from a representative individual of each species. The researcher then calculated the percent divergence in the sequences between each pair of primate species. And you see this table 1. So you see we have the human, the gorilla, the orangutan, the gibbon, and the chimpanzee. And you see we have the same ones across the top. The way that we read this is we see where they intersect. So the human intersects with the gorilla. That tells us that there's 10.3% differences. The human and the orangutan have 16.1. The human and the gibbon have 18.1. The human and the chimpanzee, 8.8. And you can do the same thing where gibbon and chimpanzee is 18.9. So you kind of like see where these two intersect. The reason why there's nothing written down here is because it's a mirror image. So you will always see these charts like this where it's just going to have like a diagonal with dashes because the human and the human, of course, have no differences. And then you'll see the number of differences usually on the top part of this. And then the bottom will be kind of be shaded to say, hey, these are the same. Don't worry about this. Okay. So in part A, it says, based on the fossil data, the researcher estimates that humans and their most closely related species in the data set diverged approximately 7 million years ago. Using this data, calculate, there's math, the rate of mtDNA, so the rate of that mitochondrial DNA, percent divergence per million years between humans and their most closely related species in the data set. And then we'll round our answer to 2 decimal places, okay? So which one is most closely related to the human? Okay, so we have to look at which one has the lowest percent. Okay, so if you look across that row of human, the gorilla has 10.3, orangutan 6.1, gibbon is 18.1, and look, chimpanzee is 8.8. Okay, so we can see that the most closely related species with the human is the chimpanzee with 8.8. Okay, now they said that we're looking at that this is taking place over 7 million years, right? So we take that 8.8 and we divide by that 7, which gives me 1.257. And then they asked us to give that to 2 decimal places. So since I have a 7 here, the 7 causes the 5 to be rounded up, so I get 1.26. They did give you a little bit of leniency, and if you put 1.25, you would have still gotten credit in case you don't know how to round, and you just drop that 7. But the answer should have been 1.26, okay? So Simple math, nothing too crazy there. It was just a proportion, right? Because remember, a lot of the math that you're going to see is just them being complicated, right? They're going to tell you something you've never seen before. And most students have never had to do that math before. But it was very simple. All you had to do was look at what was the differences and then divide by the amount of years that they said. They said it was 8.8 differences, and they said it was 7 million years. So just divide it to see how many it is per million years. Okay. So the next part of our thing is that we need to use the data from the table to construct a cladogram on the template provided, and then provide reasoning for the placement of the gibbons as the outgroup. Okay. So whenever we're looking at these cladograms, okay, they always will give you the graph, okay? Ever since the 2013 redesign, we've always been given the blank tree or the blank cladogram, and all you have to do is fill it in. So all you have to do is understand how to put the names down on that graph, on the chart, right? So we already said that the human and the chimpanzee were closest related, okay? So we're going to put those down first, okay? So we put the human and the chimpanzee. Now notice there is a branch point right here. Okay. Branch points mean that they can be rotated. So if you put the chimpanzee first and the human second, you're perfectly fine. You're not wrong. You are perfectly fine. There's no issue at all. Oops, I pressed button too soon. Okay. So now we have to look here and say, okay, well, which one is next? Okay. So again, let's compare that to the human because it's all in a straight line. It's easy to follow. So the chimpanzee was 8.8. The next number we see that's going to be the highest number will be 10.3. So we then put the gorilla on the next line. Okay. And then we say, okay, what's the next highest one? Oh, that's 16.1, which is our orangutan. So we put the orangutan over there. And then they've already got the gibbon on the graph for us. Okay, so easy peasy. All we had to do was just look at it, right? And put them in order based on the number. Now, a common mistake the students make is they'll just put them in order of the labels, okay? Now, notice they have specifically put them out of order in terms of the numbers just to ensure that you understand how to place them. So again, we're going to start with the ones that are the lowest number, and then we're going to build up from there. So the human and the chimpanzee were first because they had 8.8. Then we went to 10.3 with the gorilla. And then we went to 16.1 with the orangutan, and then we went to 18.1 with our gibbon, okay? So, they said provide reasoning for the placement of the gibbon, okay? So if I look at that gibbon, okay, it has 18.1 differences with the human, 18.9 differences with the gorilla, and 18.9 differences with the orangutan, and then 18.9 differences with the chimpanzee. It has the highest number of differences from any of the other organisms, okay? And so that's what you have to say, is that it has the least similar, it's the most different from them, or you could say that it's the most divergent. It has the most differences in terms of its DNA, so it has diverged soonest or earliest in the evolutionary time, okay? So the next thing we have is on the cladogram, draw a circle around the species that are descended from the species indicated by the node in the square. So right here, sorry y'all. Right here, we have a little box, and they want to know which one's descended. Okay, so I always like to think of it like you're driving down a road. Okay, so as you drive down this road, you then took a turn and there's gibbon. Have we reached the box yet? Nope. Keep driving. Okay, we turn her over. Oh, look, there's a orangutan. But once we reach that box, everything after that box has been on that road, right? So the gorilla, the chimpanzee, and the human are all descendants of that box. So we would have put a circle around those three organisms, okay? So that was our short FRQ. Nothing too crazy there. Nothing too, out there. And then the next FRQ we have is a long FRQ. This is the one that we just did in class. And again, I'm talking to my students. We just did this one today in class. I already gave you your paper back, but we didn't have a chance to talk about it because I was too busy grading them all, right? So I wanted to make sure that I went over this one in case you needed this, okay? So what they did is they gave you a big phylogenetic tree. Okay. And they say polar bears are highly adapted for life in cold climates around the North Pole. Brown bears, black bears, and pandas are found in warmer climates. Research collected complete mitochondrial DNA sequences from several populations of bears and constructed a phylogenetic tree to represent their evolutionary relatedness. Okay. And so here you see, of course, that tree. So the researcher is studying adaptation in bears, sequencing the nuclear gene, encoding a lysosomal trafficking protein list in polar bears, brown bears, black bears, panda bears. There are 7 inferred amino acid substitutions that are found only in polar bears. Mutations that cause similar substitution in the human list protein are associated with a syndrome, autosomal recessive condition in which pigment is absent from the hair and the eyes. Researchers then inferred amino acid sequences to build the distance matrix. So here you can see the matrix of the number of differences in those amino acids. Okay. So using our tree, we need to estimate the age in hundreds of thousands of years of the most recent common ancestor. So when we look here, we want to say the most recent common ancestor of bears. So here I have the European bears. Here I have the Western North American brown bears. And here I have the Asian brown bears. So where do they come in contact together? Right there. If you follow all their little paths back, this is when they first come together. So we drop a line down and we say, okay, well, where is that? So that's I don't know, 3.2 maybe. So you would say somewhere between 3 and 3.2, right? And then you say hundreds of thousands of years. So now students had a lot of trouble with this on the actual exam. I didn't grade this one at the real exam, at the real read. But I know that there was a lot of confusion, right? You saw hundreds of thousands of years and students put some unknown number of zeros between it. Okay. So for the scoring guideline, they were just very simple. And they said, as long as the number that you got is between 30 and 35, you got credit. Okay. So whatever number you have, as long as it's somewhere in that ballpark between 30 and 35, 3.0, 3.5, as long as you have those digits, you got credit for this one. Okay. So we then had to identify the brown bears in which polar bears are most closely related in mitochondrial DNA sequence comparison. Okay. So, we need to look at our polar bear and say, okay, well, which one's most closely related? So if you notice directly right, I'm using my wrong hand for this. Okay. Right here, we have a branch point, right? So that shows that the European brown bears are most closely related to the polar bears. Okay. So that would be the European brown bears. Okay. And then we have to identify 2 populations whose positions could switch without affecting the relationships illustrated in the phylogenetic tree. Okay. So anytime you have a branch point, they can rotate. So you can talk about European brown bears and polar bears could be switched. You could also say that our Western North American brown bears could also be switched with our Asian brown bears. Okay. Those ones could switch. You cannot switch these 2 because where that branch point takes place is in a different location. So we would be unable to switch these on this diagram. So Jessica's asking if it wouldn't count. That would be perfectly fine if you put 320,000 or if you put 330,000. You can't put the or in your answer because you're not giving a definite answer. But if you'd written either of those numbers, you would have gotten credit. So we now have to construct a cladogram based on the template to represent the model of evolutionary relatedness. Okay. Again, they've given us that tree, so we are easily able to just fill it in. Okay. So circle the position on the cladogram that represents the outgroup. So first thing I have to do is I have to look at the graph. Okay. Or look at my data. Now here's another one where they did not put in order. Notice where is our lowest number of differences? Where is the lowest number that we see? Well, you see a number 1 right here, and that's between our black and our brown bear. So between our black and brown bear, there's only 1 difference. So that means that we're going to put these 2 together on our tree. Okay. Now again, you can switch them. That could be black and that could be brown. It doesn't matter because there's a branch point there. Okay. But I just went ahead and just put brown and black there. Okay. The next thing we say, okay, well, where is our next number of differences? Okay. Well, if I look, I see there's a 7 and an 8 with the black and the brown bear with the polar bear. So I know that the polar bear is next. Okay. Then, I need to address that question in a second because Ryan's not correct. So then in our next thing, we see that this is the highest number of differences. So the last one we're going to see is our panda bear. Okay. And so then we need to circle the position on the cladogram that represents the outgroup. So the one that is the first that diverged off right here, that is your outgroup. Okay, so you should have had black, brown, polar, panda, and then you should have circled this last one on the outside. Okay, so now in the chat, Oli is asking if you wrote 300,000 to 350,000 and put a little dash in there, if that would be correct. That is incorrect. Okay, you have to give a answer, a definite answer. You cannot give them ranges. You cannot say it's this or this. Give the readers one answer. Because when you give them multiple answers, you're just showing that you don't know the answer and you're just trying to hope that you get a point for something. Okay. So you need to always be definite. Don't give me never should there be an or in a sentence when you're trying to give them an answer. Okay. So be very, very careful that you don't give them ranges. Don't give them ors. It was 320,000. It was 330,000. It was 350,000. Whatever number you're writing, give me one number. Okay. So the next part of our question says, student claims that mitochondrial DNA sequence comparisons provide a more accurate phylogeny of bear species than do list protein sequence comparisons. Provide one piece of reasoning to support the student's claim. Okay. So we mentioned this earlier, right? DNA is going to have differences in it that may not be seen in your protein, right? You can have a difference taking place in a silent mutation, right? You change that one nucleotide, but it still has the same amino acids. So you don't see a difference in the protein that occurred within your DNA, right? You could also have the mutation taking place in an intron or in a non-coding region. And so since these are not coded, they're not part of the amino acid structure, so you won't see those differences. Or since intron, it gets cut out in post-transcriptional modification, again, you won't see it, so thus it's gone, right? Another thing is that if you're looking at the DNA, you're seeing multiple genes versus when you look at the protein, you're looking at a single gene. So that's another advantage that we see. Okay. And then the last part, it had on there was, phenotype associated with the list gene is under strong selection. That's more or less talking about natural selection and talking about that the gene is selected for in a certain trait. Yeah. Okay. So D, a researcher genetically engineers a mouse strain by deleting the mouse's list gene and replacing it with a polar bear list gene. Predict the most likely difference in the phenotype of the transgenic mouse strain compared to the mouse wild type. Okay, well, we know that in the polar bear has white fur because it has that list gene. So if we take the mouse gene out and we put in the polar bear gene, you would expect that the mouse is going to be white because it has that gene that caused that pigmentation. Okay. And so that would be your answer is mouse is white. And that would be your reasoning because in the polar bear, it caused the mutation caused there to be white fur. Okay. You could have also connected it to, the human gene. So in humans, it caused the, was it absence of pigmentation and absence of in the hair and the eyes? I believe is what the prompt says. So that's what you should have had on your answer is you should have talked about that the mouse fur and or eyes would not have the pigment or would be reduced pigmentation. The mouse fur would be light or white. It had to do with that polar bear has the associated with lack of pigment or white hair, and the mutation of your human gene is associated with the lack of pigment in the hair and the eyes. Okay. And the last thing we have is describe how the mutation became common in the polar bear population. If the list was okay, that's the second part. Okay. So why did it become popular? Well, number one, natural selection. Now it says to describe. So you can't just say natural selection. That's called word dropping. Okay. Where you literally just name drop some phrase. You're like, yeah, this is natural selection. And you just drop it. Okay. Well, that doesn't tell me anything and you didn't describe anything. So you need to describe it, right? So the polar bears that had the mutation and had the white fur were more likely to survive. And because they were more likely to survive, they were more likely to leave more offspring, and those offspring were able to survive as well and continue to have offspring. And so we saw this list gene increasing in the population. If you want to add in that the reason they were able to survive is because their environment is very white, so they were able to camouflage, you can add all that other fun stuff in. But the big thing you had to have was they were more likely to survive and more likely to pass on that gene to their offspring, who are also more likely to survive. I don't think Mendel's law is on the test. Let's talk about which. Okay. So the second part of the question says to predict the percent of white offspring produced by the mating between a polar bear and a brown bear. Well, it's only found in it's only found in polar bears. And so we're looking at a homozygous dominant with a homozygous recessive, which will give you 100% heterozygous, which will not be white. So you would expect there to be 0% that are white. So with that is all that I have planned. Okay. Do we have any questions for your test tomorrow? I will give, quick little moment for you to, whatever questions you have, do you have them? I don't see anything that's been put in the chat box in terms of a question. I haven't seen anything in the question box. Do you need anything else from me before I sign off, since you have your test tomorrow? Test has 30 multiple choice questions, 4 numeric responses, although one of them isn't really a numeric response. 1 long free response, 3 short FRQs, same as normal. You got 50% is multiple choice and numeric response, 50% is your free response. I think I've killed enough time. No one's asking questions. So remember, AP Bio Penguins, you're not getting candy canes. AP Bio Penguins are dressed for success. Good night, y'all.